that overexpress the EGFR, epidermal growth factor receptor. So that's one of the things that we can tease out by studying both the human cancer, identifying those genetic changes, and then introducing them into our mouse models so we better understand how that particular mutation affects either the development of cancer or the prognosis afterwards, or even to identify different targets for therapy. So because we can engineer the mice, as I said, the designer mice, we can also create mice that express these specific mutant genes. We can do this in a variety of ways to experimentally manipulate a particular target that we found to be important. So here's an example of mouse models that allow us to analyze these particular kinds of genes. This is my favorite protein, which is called TGF-beta. And one way that we know that TGF-beta is important in breast cancer is by the early studies by Charles Daniel and Gary Silberstein, where they took little pellets, kind of like time-release capsules, implanted them in the mammary gland, and showed that as a consequence, the mammary gland regressed. So this indicated that this transforming growth factor was actually very good at inhibiting growth. So it wasn't really surprising that many tumors escape the regulation of TGF-beta so that they can grow in this uncontrolled fashion. But further mouse models were able to better demonstrate that. For example, in one mouse model done by Pierce and colleagues, they overexpressed TGF-beta so that it was constitutively active. And again, they replicated the effect of the little LVAX palette and showed that they actually got reduced ductal development and inhibition of cancer. So that's great. Here's a molecule that will actually inhibit growth locally and inhibit the eventual development of cancer. So what happens if you now take and express this at one point in the animal's life versus another point in the animal's life, later in life, so that it only expresses during lactation, is you find another feature of the TGF-beta molecule, which is that it inhibits the ability of cells to make milk. So now we're in a kind of quandary. We have to now dissect out when you want to inhibit TGF-beta. Do you want to inhibit it early so you don't have very much ductal development? But if you do so, then you also prevent the woman from undergoing full differentiation during pregnancy, and so she can't suckle her children. So you have to weigh those balances. And this is one of the ways that we continue to work on this protein for a variety of perspectives. Now, another aspect of breast cancers and normal mammary biology is actually that tissues, those complex little ductal trees that I told you about, are composed of individual cells. And we're very interested in the composition of the ductal tree. We want to know who's doing what to whom, which part of the tree is important, which cells in the tree are important, and in particular, we want to ask the question, what is really the target of cancer? What I mean there is, it's which cells are susceptible to giving rise to cancer. So in order to ask that question, we have to now zoom in a little further and look at the composition of the mammary tree. And we do this under kind of a rubric. We call this, so we have the genome, which is the DNA sequence. The phenome is how the DNA sequence is expressed in any given cell. Since overall, as long as you don't have a tumor, every single cell in your body has the same DNA sequence, but it's being expressed in a variety of different ways, so that you have skin cells, and you have a lens in your eyeball, and you have a brain, and you have breast. That's the way the genome is being played, kind of like a piano, the difference between Mozart and Chopin. So here we have different tools that we now apply in order to map the particular functions that are being expressed in a given cell. We can do this by localizing proteins, and we can also use it when we go to characterize tumor types, because again, we want to know whether the tumor is ER positive or ER negative. So here's an example from my laboratory where we're looking at mouse mammary gland, and what we're looking at here is now cross-sections through portions of the ductal tree. In one case, we've stained the nuclei, which contain the DNA, with a red dye on the what side is that? Left. And over here we've stained it with a different colored dye, which is stains the nuclei blue. One case, what we're looking at is an extracellular matrix protein called laminin, which is shown now in green. So you can see this little ruffled color around the epithelial nuclei and how exquisitely specific you can get in terms of localizing a particular protein to a particular place in a particular tissue. On the other hand, we can also look at the features of individual cells within the epithelium. Here, again, we have the epithelium of the mouse mammary gland, and what we're looking at here is this very important marker, the estrogen receptor, which receives the signals from the ovaries to induce proliferation during normal mammary gland development. So we're very interested in what regulates the expression of estrogen receptor because of its role in the development of breast cancer. So this is just one way that we can map specific functions to particular cells. Why do we care about some proteins like laminin? As I showed you back here, actually laminin doesn't sit within the cell, it sits outside the cell. And part of what we've learned by mapping all these proteins is that actually it's the interaction of the cells with its microenvironment that tells the cell what it should be doing at a given time or at a given place. So what do I mean by that? I mean, again, so in the mammary gland, we have myoepithelial cells, and we have luminal epithelial cells, we have fibroblasts, and we have inflammatory cells. All these cells know what portion of the DNA that they should be expressing or playing at that particular moment in time because of two things. One is their history. So a mammary gland develops from a particular portion of the gland, of the embryo, and because of that development, it knows it should be a mammary epithelial cell. And then the second set of signals comes from the microenvironment. So in this cartoon, we start off with a single cell, but then we have multiple cells, and those cells actually attach to each other, and that's one set of signals. Those cells also then sit down on this kind of specialized protein membrane called the extracellular matrix, of which I just showed you an example of, laminin. And by sitting down on that particular kind of membrane, the cells receive another set of signals. Together, this extracellular signaling and intracellular signaling on the interior of the cell tells the cell what it should be doing and how it should behave in response to specific stimuli. But we're still not complete yet, because right now all we have is a couple of cells together. We don't have cells organized in anything that's appropriate for example, the production of milk. You need a third component, which is morphogenesis, the organization of cells into a functional unit that then allows differentiation to occur, and under, again, the appropriate extracellular signaling. What I've schematized here is, again, a little cross-section of those ducts so that these cells can now make milk. So these breast cells are residing in a specialized microenvironment, and by looking at the details of how they're organized, again, here's just laminin in what I just think is a really pretty picture, we can learn more about how we can recreate and study these cells. So if you take cells out of a mouse or out of a woman, a person who donates tissue to biology, you can separate the cells into individual components, fibroblasts or epithelial cells. Here we've taken the epithelial cells and we've put them in a traditional petri dish, plastic actually, plastic with a particular charge on it, but plastic nonetheless. Now, nothing I've shown you thus far looks like plastic in the human. It's rigid. It has no protein membranes laid down on it. And so, actually, the cells don't look like much of anything I've shown you before either. They look flat. They look flat. Not what an epithelium should look like. In contrast, if you take those same cells and now put them on a specialized matrix, which in this case is called collagen gels. We've all heard about collagen if you read all your literature coming with your face creams, right? Collagen is what keeps your skin supple, but actually there's a huge proportion of the protein in your body is made up of collagen. And by providing these cells, now still in a petri dish, but with a layer of collagen underneath them, they become much happier looking, round, plump. They have these nice little speckly bits on their surface, which are the little fingers that allow them to secrete milk into the extracellular space. But we can go one step further. We can give them, instead of collagen, remember I told you that the cells in the mammary gland sat on a very special protein called laminin. And when we take those cells, these cells, and put them on a laminin, which is a reconstituted basement membrane, the things the cells actually sit on in vivo, then we can recapitulate even greater the morphogenesis of the mammary gland. So actually now we've gone from these two are about the same scale. Now we've gone to a much lower magnification, because now what you're looking at is about 1,000 cells. And the reason why it's blue in the center is because what you're looking at is a hollow sphere, that these cells have organized themselves. They were flat and simple to begin with, single cells, but you give them this extracellular matrix, now they organize into the appropriate tissue unit for the mammary gland, which is a hollow sphere where they actually secrete milk proteins into the center of this. And we can see how closely they resemble the cells in vivo by comparing a cross-section through this culture in this little, we call them mammospheres. Cut in half, we can now see that you have a simple epithelium with the cells oriented towards this lumen. There's a little bit of debris in there that remains from their organization, but it's very similar to what actually a mouse mammary gland looks like when it's making milk protein. So here again, the simple mammary epithelium, and in this case, the milk proteins in the center of that. So by observing how the tissue is organized in vivo, we can now begin to create better models of how we should get the cells in the right orientation, so we can now study the response to particular events like the exposure to ionizing radiation. Kind of one way to put this into perspective is that we tend, as biologists at least, to think of cells as a unit of function. But tissues are no more a collection of cells than a house is a collection of stones. In other words, you can't just reassemble bits and pieces of, by using, or you can't reassemble a full mammary gland just by thinking of cells as individual units. You have to think of it as a system and provide all these systems. So you have to have cells that proliferate, so you have to have a lot of cells, but you also have to give them the right cues so they undergo this tissue-specific morphogenesis. And in doing so, we can then achieve a functional cell culture model that we can manipulate. Likewise, you can use the same property to discriminate between normal cells and tumor cells, because a tumor cell proliferates just fine, but it loses the capacity to organize. And so one of the characteristics of a tumor is a loss of architecture, so that it no longer organizes into a functional unit and loses the kind of constraints that are placed upon those functional units, and begin to invade in the surrounding tissue and even move to distant sites. So it takes a tissue to make a tumor. You can't think of cancer as being a disease of cells. You have to think of it as being a disease of tissues, and you have to think of it as being a disease that is expressed as a function of multiple cells. So we have to begin to think about how you go from this nice normal mammary tree that has all its cells orderly lined up to this mass of unorganized cells. So what's required to make a breast cancer? First of all, you need the epithelium. I've already mentioned that in my first cartoon, that target cell is epithelial. It means that when we're talking about breast cancer, we're not talking about fibroblasts, and we're not talking about inflammatory cells. Those are different kinds of tumors. For breast cancer, the target and the seed that begins the whole process is an epithelial cell. However, that seed needs to be placed in the appropriate soil. And that soil is actually the surrounding cells that I mentioned earlier, the stroma, the fibroblasts, even